Okay, so today I'm talking about enzyme structure and function. So just kind of what a protein is, what it does. We've talked about how it catalyzes these reactions. We talked about the fact that it is a protein, but kind of how does it do that job? And then tomorrow I'll be talking about different types of protein or enzyme inhibition. Okay, so today enzyme structure and function. Okay, so usually when we talk about enzymes, you have most of the enzyme and then you have a shape. Now it isn't exactly like this shape, but it'll have what is called an active site. So this is called the active site. And this is going to be where the substrate binds to the enzyme. Cool. So kind of active site, substrate. Substrate is the actual thing that the enzyme is breaking apart. And so for the enzyme I drew, it's going to look something like this. So it's going to have two parts and it'll be rounded here. Kind of color that in. And then it'll have this other part that matches here, right? So see how the shape matches? So what will happen is the substrate will come into it. So I'm just drawing like a little half circle and a triangle. The triangle part gets filled and it fits perfectly in there into the little spot of the enzyme. So then what happens is when the substrate leaves the enzyme, it leaves as two products. So products. Write that there. So I have the active site, but then I have my one product over here, and then the blue triangle thing over here. Okay, so it goes from we have the substrate in the enzyme, the shape has to fit like a lock and key type thing, and then it breaks it apart. And so kind of, I think this might have something to do with hydrolysis. Let me read for a second. So, and one thing to note is that this will be, so this is substrate entering the enzyme. And then that would be at the active site. Active site. And then in the middle, there's really two steps here. So if we were to imagine that our substrate actually looked like this when it entered the enzyme. So for time's sake, I'm just going to draw the bottom here. But it just looked like that, and then so this is the same step as this, but then that moves along to the substrate enzyme complex, or enzyme substrate complex. So still blue and red, but in it. And then the next step it's going to be is going to be the enzyme product complex. So as you can see, we're moving towards the this final step here. So I'm going to plot my other two colors again. And for this diagram, I want you to notice how the enzyme, the substrate's been broken apart into the products, so that's why we have a product complex. but still is in the enzyme. And then we just have products leave. So products leave. That's going to be this step. Oh, nope. This step. And I'm basically going to draw the same thing twice. That in blue. So that's the basic process. And then one other thing to note is that enzymes usually have an ending, the ending of ase. So like lipase, proteinase, DNase, kind of things like that. And so it corresponds to that. It's going to be fats. It breaks down fats. It breaks down protein. This breaks down DNA. Different things like that. And it breaks things apart. To lower the activation energy. So that's kind of the ins and outs of how an enzyme works. And next will be how enzyme activity, this process, can be inhibited. Hope that was helpful.